Hi there, first of all I want to thank ASBMB for giving this opportunity and making this work during these very chaotic times. My name is Jake. I recently completed my PhD studies at the University of New South Wales in Sydney, Australia. Now I'm a fresh postdoc at the Water Analyzer Home Institute in Melbourne, Australia. Thanks to Andrew and Becky for allowing me to present this work today. The work I'll be presenting is from a PhD. All the data shown here have been published. I want to sum up about 3 to 4 years of work by giving you the main message and concepts rather than overwhelmed with data. My initial poster and oral presentation sessions for EB 2020 was regulation of lipid metabolism. You can get a copy of my poster at this link here. So I work on Squalene Monooxygenase, or also known as Squalene epoxidase, a rate-limiting enzyme of cholesterol synthesis. It sits here in the long schematic of the cholesterol synthesis pathway, and here in the simplified version catalyzing this step. This is the structure of Squalene Monooxygenase. It has a catalytic domain and a regulatory domain, which is my focus. Interest in targeting human Squalene Monooxygenase for the treatment of hypercholesterolemia started and ended around this period. In recent years during my PhD, new interest in SM, exemplified by some papers, emerged in the cancer metabolism field. My PhD focus was to understand how this enzyme is regulated. Given we knew little about it at the time, from my PhD advisor's lab, they showed when there is excess cellular cholesterol, SM is degraded rapidly through the ubiquitin proteasome system, shutting down the cholesterol synthesis pathway, and this represents a nice feedback mechanism to regulate cholesterol synthesis. This response requires the first 100 amino acids, a degron region, which means a degradation signal. If you lose this degron, SM does not undergo this cholesterol-mediated degradation. You can study this degron closely in isolation by transferring it to GFP, which imparts this cholesterol responsiveness, and I think it's quite remarkable given how short this domain is. To conclude, the domain is important as it ultimately controls the amount of Squalene Monooxygenase catalytic activity at any given time inside the cell, thus impacting cholesterol synthesis. The lab showed that a re-entry loop exists within this degron and undergoes cholesterol-induced conformational changes around the borders of the re-entry loop, a work published by a colleague, Dr. Vicky Howe. From my PhD work, we've painted a more complex picture which I will discuss today. Early on, we asked some simple questions. How does the degron sense cholesterol? Are there motifs or specific amino acids needed for this? We have done truncations from the C-terminus of our SM N100 GFP degron. We express the mutants in CHO7 cells and treat them with cholesterol. SM N100 is localized to the endoplasmic reticulum in these cells. Wild-type SM N100 with cholesterol is degraded. These degron truncated mutants show a lack of cholesterol-mediated degradation, indicating this region might be sensing cholesterol. Long story short, after reading about a yeast degron, we introduced 4 mutations across our degron, which remarkably blunted this cholesterol response in the SM N100 degron and full-length Squalene Monooxygenase. Perhaps these 2 mutations affect the re-entry loop, but why these? Now, these 2 residues lie within the truncated cholesterol-resistant mutants earlier. We also did systematic mutagenesis, mutating every second amino acid to alanine. At first it was fun, but after 200 plates of transformation and a lot of Western blots, it was slightly traumatic. We had a bunch of negative results, but at least we ruled out some motifs. Perplexed at the time with the 4 mutations, we were interested in this unstructured region. But from prediction programs, we identified an amphipathic helix, and we could show with circular diachirism of a peptide for these 12 amino acids that a helix does form in the presence of detergent DDM to mimic a membrane. Without detergent, the helix disappears. Upon deletion of this helix, you can see a remarkable loss of cholesterol regulation. This led to a series of experiments and a publication on the amphipathic helix sensing cholesterol. So refining the degron model, cholesterol-mediated unfolding triggers an alert signal for degradation. But successful degradation needs ubiquitination of proteins. We already knew that MARCH6 and E3 ubiquitin ligase adds ubiquitin on this degron. So where are the ubiquitination sites on this degron? Ubiquitination usually occurs on lysine residues, but from earlier work, the lab showed a lysineless SM N100 degron was still degraded by cholesterol, indicating it's likely not lysine ubiquitination. There are reports of non-canonical ubiquitination on serine, threonine, cysteine, but uncommon. We next mutated all potential non-canonical residues in clusters and in combinations, as seen in the table. Perhaps the E3 ubiquitin ligase comes in to modify the protein at one site, and if it's not available, it goes to another site. By this point, you might think it's only 100 amino acids. How hard can it be? At the end of my PhD, we've got more than 100 mutants for this SM N100 degron. We could see a lot of mutants that lost cholesterol responsiveness and have higher protein levels, but just which residues are likely to be top priority for ubiquitination. Carefully quantifying our Western blot and analyzing our data, we narrow it down to the second half of this SM N100 degron having multiple serine ubiquitination sites. Most of our Western blots are quantified, giving us statistics and confidence in our results, especially with these kinds of mutants having a spectrum of response. Mass spectrometry could show serine 83 is indeed ubiquitinated. We also have other indirect evidence for the role of non-canonical ubiquitination from other experiments as listed here. Building on our model, we now have an eat me signal that unfolds with cholesterol, allowing serine ubiquitination within the nearby regions, which requires UBE2J2. Finally, this hydrophobic degron must exit the endoplasmic reticulum. Our lead candidate for an extractor was valosin-containing protein, or VCP, which is a quite conserved element for endoplasmic reticulum-associated degradation, or known as ERAD, in these mammals. From pharmacological inhibition of VCP by CB5083 and siRNA-mediated knockdown of VCP, you could see both SM and SM N100 could be stabilized substantially here, 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 and here. Furthermore, the cholesterol-mediated degradation could be blocked here, 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 and here. Coming back to this, we now have an extractor, valosin-containing protein, that allows exit from the endoplasmic reticulum membrane for proteasomal degradation in the cytosol. Despite being around for ages, it was only last year that the catalytic domain structure was solved. To reiterate, the activity needs to be regulated by this degron. Our laboratory has worked substantially to understand how this degron executes its regulatory actions to control Squalene monooxygenase abundance, which effectively regulates cholesterol synthesis. This degron is actually able to sense other lipids too. I focused on cholesterol sensing, but we and others have described other lipid sensing roles. Early on in grad school, I used to perceive membrane proteins as having transmembrane domains only, but later learned of these other structures that pin membrane proteins down, the loops and amphipathic helices, which have effects on protein function, localization. For example, lipid droplet proteins are characterized by these hairpin loops and amphipathic helices. Squalene monooxygenase is actually found on lipid droplets too, but why it's there or how it gets there is actually not known yet. Our findings are quite exciting because sterosensing or sterobinding is usually thought to occur through sterosensing domains like those found in SCAP or HMG co-reductase. These sterosensing domains are transmembrane domains. There are also crack and carp motifs that allow cholesterol recognition in some proteins. However, our sterosensing mechanism is probably more similar to sensing of membrane properties in response to fluctuations of membrane cholesterol content. A good example of membrane property sensing is lipid packing defects sensed by the amphipathic helix of CCT, a rate-limiting enzyme of phosphatidylcholine synthesis. We've also got non-canonical ubiquitination, which we propose is facilitated by unfolding of the helix with high cholesterol. This adds to the small list of substrates ubiquitinated on serine, threonine, and cysteine. One other example of non-canonical ubiquitination for a cholesterol-related protein is ACAT2, and perhaps we can expect more examples to come in the future. What we discovered with Squalene monooxygenase cannot be done without the scientific discoveries provided by other researchers in their respective fields. Squalene monooxygenase isn't the only example where lipid homeostasis is intertwined with protein homeostasis. The endoplasmic reticulum is a busy organelle for lipid metabolism and protein quality control, and others have discussed this relationship between metabolism and protein quality control as well. Lastly, I'd like to thank the organizers again, and I wish to thank the members of the Brown Lab, and also my PhD advisor, Professor Andrew Brown, the collaborators, and lots of other people who made all this possible, and thank you for listening to this presentation.